Given the timing, I might be moving a little bit fast, but all of the slides as well as citations are going to be available at this link down here, and this will show up again on the thank you slide. So if I go through anything too fast or you're interested in learning more, please don't hesitate to follow up. So just a brief outline of what I'm going to talk about. I'm going to give you a little bit of background on the links between the gut microbiome and the brain, tell you a little bit about two cohorts of child development that I'm privileged to be able to study to answer questions around this, and then finally give you some of our initial results linking gut microbes and their genes with cognitive development in kids. So first, just a little bit of background. As Ruminococcus already described pretty well, we know that the gut microbiome development begins at birth and progresses quite rapidly over the first years of life, achieving a sort of adult-like microbiome by about 3 years of age. And of course, the brain also develops quite early in life and has a critical early period of development. The brain development and the production of neuronal synapses begins before birth and really accelerates just after birth and in the first years of life. So that by the time a kid is 3 or 4 years old, a lot of brain development and a lot of the patterns of neuronal connections have already been established. There are many mechanisms that link the gut microbiome and the brain through what has been termed the gut microbiome-brain-axis. So this is direct signaling between the gut and the brain, both through the nervous system itself, as well as through endocrine signaling and even signaling from the immune system through cytokines. And then, of course, the microbiome can affect the gut directly through the production of small molecules. Ruminococcus talked about a bunch of these already. This includes things like short-chain fatty acids and lipid polysaccharide, but also microbes, it turns out, can produce neurotransmitters that can directly act on the enteric nervous system, which can feed back to the central nervous system. And they can also metabolize both producing and degrading neurotransmitters that can enter circulation and cross the blood-brain barrier directly. So there's a lot of potential links here. And of course, we're interested in the microbiome, which, as distinct from the gut and the brain, is uniquely accessible and manipulable in principle. So the questions that we're interested in asking in our lab here at Wellesley are, what role, if any, do microbes play in typical human neurodevelopment? Most of the work that's been done to date has been in neurological disorders. And we're interested in really what's happening in healthy children with normal development. Which microbial species play these roles? And of course, also as Ruminococcus mentioned, this can be context and location specific. So just focusing on the developed world is probably not a great idea. And then finally, what microbial genes and metabolic activities mediate these effects. So the primary cohort that I'm going to talk about today is called the resonance cohort of child development. It's based just a little bit south of me in Providence, Rhode Island. And this is an accelerated longitudinal study where we're collecting stool that we send for metagenomic sequencing, so we can get both taxonomic and functional profiles. And kind of uniquely for this study, we're also collecting neuroimaging for many of these kids as young as 3 months old. So we actually stick these kids in the scanner and we can get really detailed images of their brain structures. And in addition, we also collect assessments of cognitive function that I'm going to describe in just a sec, a little bit more. And we feed all of these modalities as well as the sort of typical sociodemographics into a number of different kinds of analysis modalities. The other cohort that I'm not going to present any data for today, but that I'm really excited about is called the Kula cohort. That means grow in the local languages. And this is a similarly designed study where we're collecting longitudinal samples, stool samples from these kids for both metagenomics and metabolomics, as well as doing neuroimaging with MRI and EEG, and collecting a lot of other measures about these kids' development. So no data on this, but stay tuned. So as you might expect, the microbial metagenomic profiles that we collect are highly age dependent. These plots are all ordinations where in the dark colors, you're seeing the youngest kids in the study. And you can see that for both functional profiles and taxonomic profiles, and also for neuroimaging profiles, you can see that the youngest kids are sort of in a class by themselves, which can be challenging for some kinds of analyses because a lot of things are age dependent and it's hard to disentangle age from the statistical effect we're looking for. But the cognitive function scores that I mentioned briefly before have the advantage of being mostly age invariant. So we use a number of different kinds of cognitive assessments looking at different aspects of child development, from motor learning to language acquisition. And the nice thing about these different studies or these different kinds of assessments is that they can all be normalized to a common scale. Here we're normalizing to an IQ-like scale with a mean of 100. So because all of the microbiome features that we're interested in are age variant, it's nice to have at least one thing that doesn't vary with age, so it's easier to untangle that effect. All right, so what do we see? If we do a sort of standard linear model approach, we identify 13 taxa that pass multiple hypothesis correction, 12 of which are positively associated with cognitive function score, one of which is negatively associated. I'll tell you the IDs of these bugs in just a moment. And in addition to looking at the taxonomic profiles, we also are looking at the functional profiles. So in our case, rather than looking at each gene or pathway independently, we used a couple of different gene sets that were identified in this publication here of potentially neuroactive genes that are grouped into a metabolism of different neuroactive compounds. In this case, the 3-carbon short-chain fatty acid propionate. And what we do is we basically do the same kind of linear model for all of the genes in the gut metagenome, and then look for the given gene set, how do they tend to be associated with our feature of interest, in this case, cognitive function score. And so this is looking at, we looked at about 20 different gene sets, potentially neuroactive gene sets, and these were the ones that had some amount of statistical association with cognitive function in the different age groups that we looked at. So after this, we decided we would hop on the machine learning bandwagon, although it actually has a number of really useful advantages for this kind of study. So one thing about many ML models, including random forests, which are what we use, is that they can identify nonlinear relationships, and they can look at the entire community together rather than looking one bug at a time. The machine learning model can actually learn which features are important, including relationships between features. And in addition, the thing that we find really valuable is separate and apart from its ability to predict some outcome like cognitive function, the models also encode which features are used in each tree split and how good the split was, which allows us to label each feature, microbial species or gene function, with its importance for prediction of a given model. And so these are the results. We use both taxa and genes to try to predict cognitive function score in kids from 0 to 6 months or from 18 months to 10 years. And what you can see is for the 6-month-olds, the models really didn't perform very well. This might be because assessing cognitive function at this age is a little bit less reliable than for the older kids. But for the older kids, we got a pretty decent prediction performance in our test sets. And then we could look at which features, which microbial species turned out to be important. And here we're comparing the results from linear models on the x-axis with the importance from machine learning on the y-axis. As promised, here are the bugs that were identified by machine learning. The ones boxed in purple are the ones that were also identified in the sort of more standard linear model approach. So we have a number of different microbes here that seem to be important for random forest models in predicting cognitive function. We're really interested in following this up. And the next slide is a bit busy, so I apologize, but here we're looking at neuroimaging. So we can segment the neuroimaging scans into individual brain regions, and we can use the same random forest approach to try to predict the size of the brain region from microbial taxonomic profiles. So here, these bars are showing the model performances. You can see some of them are not too bad. Some of them are pretty abysmal. But then again, we can identify which microbes are important for each of these brain regions. And we see a couple of interesting patterns. Some bugs are really important for a single brain region, like Blautia wexlerae here and the parahippocampus. Other bugs, like Coppercoccus comus, seem to be important for models of a bunch of different brain regions. And some brain regions have the important bugs spread out over a number of different related microbes. So the left accumbens, for example, it's hard to sort of line these up, but the left accumbens is really depends on 3 different species of Bacteroides. So I'm just going to skip through the summary so we can get to the next talks. Thank you very much for your attention. And as I said, all of the slides as well as references are available at this link down here, which I can also share in the chat later so that people can access it.